Hello, my name is Keith Ligon, and I want to thank the organizers for the opportunity to speak in this forum. What I'm going to talk about today is a project whereby we studied hypermutation as a phenotype present in gliomas. Hypermutation is defined as a high tumor mutation burden, generally greater than 10 point mutations per megabase of the genome. And previous studies had noticed that these seem to be fairly rare, but would occur after treatment, particularly with alkylating agents, but had not been able to formally establish some of the most important findings needed in order to make decisions around clinical care for these patients and how treatments might be developed. Specifically, the incidence of hypermutation in things like IDH mutant versus wild type gliomas and other subtypes was not known. The role of MMR in these tumors was not fully established, and the outcome of hypermutated gliomas, both their prognosis as well as their response rates to treatments, were not established. And this was of particular importance given the FDA had recently approved checkpoint blockade inhibitors against molecules like PD-1 in any type of cancer that has a hypermutation, MMRD or MSI. So in order to find out if these principles would apply to gliomas and answer some of these questions, two postdocs, Mehdi Tuat and Yvonne Lee, set out to collect as many gliomas as they could, which had sequencing data. They were successful in obtaining 10,294 glioma sequencing profiles. And from this, they were able to do an analysis and map the mutation burden shown here and identify tumors that had an extremely high tumor mutation burden. These hypermutated gliomas were identified and they collected a set of 558. And so this is the largest set of gliomas analyzed to date with this hypermutation phenotype. Most importantly, a high number of these tumors had full molecular and clinical information, such as MGMT status, and this allowed them to make statistically important conclusions regarding these associations. And the first thing they noticed was that overall, the rate of hypermutation in populations such as this is about 5% of glioma patients' samples had hypermutation, and again was associated with high-grade tumors more than low-grade. And recurrent tumors had a rate of about 17%, whereas primary or newly diagnosed patients untreated had a rate of only 2%. And the patients who had most frequently this hypermutation phenotype were readily identified as those who were chemotherapy sensitive and those who benefited the most from alkylator chemotherapy, specifically MGMT, DNA repair protein status for its promoter. Those patients who were methylated and MGMT deficient had a high rate of post-treatment mutation, as well as in subtypes of gliomas that were most responsive, such as oligodendroglioma, the hypermutation rate could reach up to 60% after treatment in tumors such as oligodendroglioma. Hypermutation, interestingly, was not associated with radiation therapy in our set or patients who received only nitrosoureas historically, and really was highly specific for those treated with temozolomide. And this showed a dose-dependent effect. So the patients who had multiple rounds and cycles of TMZ treatment, their incidence and risk of having hypermutation evolve in their tumor was higher, reaching a maximum of around 40% after 18 cycles. In order to determine the underlying cause of this hypermutation status and phenotype, we turned to using cosmic mutation signature analysis on the sets of HM tumors we had identified. The cosmic mutation signature looks at the context and ways in which the base pairs change and gives a specific association with prior mechanisms of mutation rates. So for instance, in patients who had never received any treatment before, their de novo hypermutated gliomas were highly associated with MMR deficiency, and this is presumably due to germline mutations such as Lynch patients who have MMR mutations. In addition, there was a second group that was defined by post-treatment hypermutated gliomas, and their signature was very unique in that it had signature 11, which has been associated and termed a temozolomide signature. But what we noticed was that almost all these patients also had a little bit of presence of MMR signature as well, or POL-E signature, indicating that underlying DNA repair deficiencies may lead to and cause the signature as well. And we noticed MMR mutations were present in almost all the cases which had this post-treatment hypermutation signature. However, because their tumor mutation burden is so high, all genes have a lot of mutations, and therefore we needed to perform a specific analysis and enrichment analysis using permutation testing in order to control for the mutation rate, the size of the gene, and things like this. And so when you carefully control for the expected mutation rate, based on those factors, what you saw was that there was a strong bias for mutations present in the hypermutated patients. In MMR genes specifically, the top mutated gene was MSH6, and also other DNA repair genes such as BRCAs were also shown to be present in these hypermutated gliomas at a disproportionate rate. And specifically, we were able to show that by IHC for MMR protein loss, we were able to confirm that 70% of these hypermutated gliomas also had very obvious loss of protein function by IHC, thereby backing up that these were functional mutations, not passenger mutations. And this supports strongly that MMRD was mechanistically underlying the HM process in both de novo patients as well as TMZ treated patients, and it wasn't due maybe to just TMZ exposure. Integrated analysis of hypermutated gliomas also identified for us that there were recurrent hotspot mutations in MMR genes. And these hotspot mutations were candidate acquired resistance mutations based on how they appeared. And so shown here, we have identified quite a number of these different mutations. You can see the most common one in this lollipop plot was the T1219I mutation in MSH6. And this occurred in 12% of post-TMZ treated hypermutated samples and had an interesting finding associated with it, which is that this had been previously published by Edelman's group and others as being a dominant negative mutation. So a single point mutation can wipe out the function completely of MSH2 and MSH6 in this setting. And so this was the strongest recurrent event seen. We next asked the question, if gliomas are now well-defined, both the de novo and the TMZ post-treatment ones as being really an MMRD disease, how do these gliomas compare to other cancers which respond with really high rates of checkpoint blockade, and how do they compare on several levels? So the first level we wanted to see was whether or not they had immune responses that might be similar to other MMRD tumors, given that MMRD has been felt to be important for triggering an immune response. So colorectal cancer with MMR deficiency, you classically see T cells marked by CD3 infiltrating into the tumor itself, and statistically you see a great enrichment there. However, there was no significant lymphocyte enrichment in infiltrating gliomas statistically in our sample sets when we analyzed this. So that was one important difference, potentially influencing checkpoint blockade. The next was that hypermutation in MMRD and gliomas was not associated with an increased indel burden or microsatellite instability as it is in other cancers. And so what you see here is comparing gliomas to all other cancers in a pan-cancer analysis for TMB. Their single base substitution rate is sometimes even higher than in other MMRD tumors, so it's not a difference in TMB. But the homopolymer indel burden was much different in pan-cancers with MMRD. Other cancers with MMRD, it was very high, whereas in gliomas, you see there are very few patients that have that as part of the process. The next thing we looked at was colorectal cancer, for instance, with MMRD actually has a better prognosis. And so we wondered if this might be the case in gliomas, but we found the opposite. So in hypermutant gliomas, both oligo and astrocytoma, we found a statistically poorer survival in those patients who were hypermutant, shown here with the dashed lines, compared to their non-hypermutant controls, shown in solid lines. And there was a trend for this present in IDH wild-type glioblastoma as well, shown in orange, but did not reach statistical significance. And the last thing we wanted to do was look at our own patients at our hospital and collect them and look and see whether checkpoint blockade, given these differences from other tumors, might show a similar response rate or a different response rate as has been reported in other cancers. And what we found was that gliomas had really a very low response rate, shown here in this graph, for the patients that we had available to be examined who had undergone PD-1 blockade. And so there was no evidence that they were achieving anything close to that, with some of the patients even showing an increased amount of tumor growth. And this was borne out also by overall survival analyses of patients who had undergone PD-1 blockade. In this instance, looking at TMB high or hypermutant versus low, we saw that they actually had a slightly worse survival than that was seen with those patients who just received other types of treatments, even when controlled for multivariate analysis. And so our conclusions were that hypermutation is most common in this chemotherapy-sensitive glioma types, and specifically associates with acquired resistance to temozolomide, but not CCNU. The dominant mutational signature of hypermutated gliomas, the signature 11, instead of being a pure temozolomide signature, we were able to determine that it actually is an underlying MMR deficient signature, so it matches the de novo patients, and as a compound signature. And then compared to other cancers, we found very striking differences in immune cells and underlying signatures and biology of these tumors that are hypermute. And specifically found that we have important implications for checkpoint blockade, and that it doesn't appear that we have high response rates in hypermutant or MMRD gliomas like are seen in other cancers, but future studies with clinical trial scale are necessary, and other indications will be important going forward to make final conclusions on that. And I'll just stop there and thank everybody in our teams at both the Dana-Farber Boston Foundation Medicine and in Paris. And thank you for your time.